Dr. Kunkel is the director of the genomics program at Boston Children's Hospital and professor of genetics and pediatrics at Harvard Medical School. Dr. Kunkel is well known for having identified the causative gene for DMD. Unfortunately, he was unable to attend this announcement tonight, and so he has also provided his video update on current research into novel therapeutic approaches to DMD using the Notch pathway.
So my lab has been interested in dystrophin-independent approaches to therapy for many years, and actually approximately a little less than 10 years ago, my lab, working with Maiana Zatz in Brazil, described a couple of escaper dogs. These are dogs that had dystrophin deficiency but lived a normal lifespan. We were able to show that those dogs had a variant in a gene called JAG1, and that allowed, and we were able to show that in zebrafish, if you overexpress a little bit of JAG1, one could correct zebrafish that were dystrophin deficient. We went on and did that in mice, and I won't talk about that, but what I would like to talk about is what we were supported by Defeat Duchenne was our work with the equivalent escaper humans, two individuals who were dystrophin deficient, who had Duchenne dystrophy, but an extremely mild disease course. Both of them were in their 20s and still ambulatory. We were able to show with Maiana Zatz, again, these were both identified by Maiana Zatz, we were able to show that they had a variant, both of them had a variant in a gene in the Notch pathway. Notch and JAG1 are ligand and receptor pairs to each other. So they both play a role in cell proliferation and cell division. And so what we did is, with support from Defeat Duchenne, we created a mouse model. So we modeled the same variants that we saw in humans in mice. We crossed that onto a dystrophin deficient MDX mouse. And what we did over the course of these last 2 years was show that those mice were improved. They had an increased strength, larger muscles. They had a whole series of different tests that we've done, all of which showed them to be much closer to normal than they were to the dystrophin deficient animals. We are in the process of putting this all together for publication, and of course we'll acknowledge Defeat Duchenne for their support of this work. At the same time, we also were starting to do induced pluripotent stem cells from the two patients that had that essentially were much more mildly affected. And we were able to show that when we got these induced pluripotent stem cells, that the patient cell lines proliferated or grew faster than did dystrophin deficient cells, and they were much more like normal. We are currently working on trying to induce, just like the variants did, induce these cells to divide and more rapidly as a potential approach to therapeutics. All of this has been supported by Defeat Duchenne, as well as an NIH grant rewarded to me from the National Institutes of Health. And I thank you all for your support.